Kia ora. My name's Dan Blanchon. I'm a researcher here at Unitec and also one of the lecturers. This particular project is in the area of biosecurity, which we are doing with Auckland Council. This plant here is climbing asparagus, one of our nastiest environmental weeds, particularly a problem in the upper North Island and in the Auckland region around the Waitakere Ranges. It's originally from South Africa and is related to garden asparagus, but this one I wouldn't eat. Its problem is that it actually will climb up native plants and strangle them. Really hard to get rid of with herbicides, and it has these little rhizomes under the ground that make it really hard to actually even pull it out. So what we're trying to do is use natural enemies to actually knock this back in the wild. So you might have known that as being a biological control. This is what we're looking at, and we're particularly after fungi. To get potential biological control agents, we need to go to the great outdoors and look for climbing asparagus plants. In particular, sick or diseased-looking plants. When we find those, we will take samples of those back to the lab and we'll work in what's called a laminar flow cabinet, which is a cabinet where we can work in a completely sterile environment. We surface sterilise with bleach, the plant material, and we dissect out any bits that have lesions or other signs of disease in the hope that we'll be able to culture fungi. Those little bits of stem we culture on agar plates with nutrient media and antibiotics, and hopefully we get something like this. These are pure cultures of different fungi. To work out what these are, we use DNA barcoding, where we extract DNA from the pure cultures, we use PCR and we amplify up a particular region, and we can then compare those sequences on GeneBank online, and that'll give us some idea of what genus or sometimes species we may have. These particular ones here, the pink fluffy one, that's a Fusarium, the sort of more orangey pink blotchy one, that's a Colototricum, and both of those have species in them that are known to be biological control agents. So that's quite promising. The last part of the process is to actually test to see if our fungi work in practice on actual plants of climbing asparagus. We put our clean climbing asparagus plant in a little tent like this one. We cut little plugs of agar with our target fungus on it, put them in the same tent as the climbing asparagus, and we hope that damage of that plant will happen, or ideally death. After that, if we've got a good candidate, we then have to test it on a range of other native plants just to make sure that it's not going to kill our valuable natives. And of course we need to test it on garden asparagus, because if we started attacking garden asparagus, we would not be popular.